about 120 students, both mainly PhD but also honours students, and we actually do offer scholarships to try and bribe you to come out there and get away from campus. So anyway, I'd like to be pleased to talk to any of you. So thanks very much.
Firstly, I'd like to thank you very much for coming along tonight. And as such, we've got something to
that's very nice.
make the trip home easier.
Thank you very much.
And also, I'd like to give a very special thanks to Luke for organising this tonight. He's put a huge amount of effort into it, and he has a real passion for it. So there's plenty more to come throughout the year. And as such, yeah, we'll continue on with questions for now, and I think we've got some pizzas coming along.
Oh, great.
So, yeah, help yourself to pizzas and please fire away with questions and use this.
So I'm very interested in research. I'm a plant molecular analysis. So it seems that your population that you're studying is dependent on the registry to form it. Maybe is that introducing bias into your sampling? Because if they have a particular allele that says, I want to participate in everything, maybe that introduces a bit of bias.
That's a good question. It's a good question. It's one that worries us all the time. You know, exactly that sort of consideration. And we've done a lot of analyses, sort of resampling analyses, weighting our sample according to different criteria, sometimes trying to actually get census data to reweight our sample exactly to census statistics on various characteristics. And what we found after doing a huge amount of work like that is for most of the things we've looked at, it makes very little difference. It might change something in the second decimal place, but we haven't found any case where it changes things in the first decimal place. But we're constantly concerned about that. I mean, the way that can happen, that bias can come in as quite subtle. So, for example, we do these twin studies, and as you will have gathered, mainly it depends upon getting both twins to volunteer. But what happens if one is very cooperative and the other's a real non-cooperative type? Then the whole pair drops out. Now, so if you leave all those pairs out where one's really non-cooperative, that's going to bias your sample. In fact, the technique we use is called full information maximum likelihood, and you leave all observations in there, whether they come from a complete pair or an incomplete pair. And what you'll see is if that trait that is causing someone to drop out is genetically determined, then even the one who cooperates will still move a little bit that way. It's a bit like what I was saying about autism, and the fact that mothers are in fact a bit colder, but they're not right out at that extreme because they've got that genetic tendency. And by including them, you can actually show through simulation studies that you are writing that bias and getting estimates that apply to the whole population. So it's a good question, but it's one that we're alert to and we take steps to do something about it. There was another question around this.
Have you ever considered looking at different model organisms for twin studies than just recruiting humans, like maybe look at twins in mice if they so occur?
Oh, I love that question. I was giving this talk to this prestigious organisation called the Human Frontiers Science Project, which has got all this money from Japan and Europe in there and so on. And at the end of my talk, which was not very different from this one, this guy actually got apoplectic. He said, you mean you're doing this research and you don't even have a mouse model? I said, well, actually, I'm quite interested in humans, I keep thinking about writing a paper on humans as a good model organism for mouse studies. Yeah. Sorry, I'm being a bit flippant, but I mean, frankly, we can do stuff in humans that you can't do in mice, and we can get much bigger registries. We've got much better genome-wide association scores. We've got much better omics in mice. I mean, where mice, I think, my frank view is that there's been far too much emphasis on mouse models in medical research. It's because that's what people do, it's what they can do, they keep pushing it. In my view, far too much of the medical research budget is diverted to mouse studies. Where mouse studies are important, I think, is when we do our genome-wide association studies and we show that genes X, Y, Z are implicated in this disease, that's the time you take those genes back to mouse models and actually try and find out what genes X, Y, Z are doing there, and then you can do all your manipulations and so on in the mice that you can't do in humans. What we've seen over the last 20 or 30 years is a huge amount of investment in people doing mouse studies, trying to make mouse models of asthma or, God help us, schizophrenia or whatever, and getting absolutely nowhere. They're finding genes in the mice, taking them into humans, and there's absolutely no effect in humans at all. And there is case after case after case of this where it's not apparent the mice are actually an extremely good model. I mean, what is it, 80 million years of evolution between them? Is that many? Or, yeah, it's huge. And it's quite conceivable that affecting the same trait, you've got quite different genes. And that appears to be the case. Anyway, that's my take on mouse models. Not very popular one. Yeah, is there a question here? Oh, back there?
I was just wondering, because you were saying that they found an 80% correlation between genetics and autism.
Yes.
And I was wondering if you knew if there was any, like, finding out what genes.
Yeah, there's a huge amount of work going on using genome-wide association studies, but what is emerging for autism and also to quite a large degree for schizophrenia, but particularly autism, there seem to be a lot of rare copy number variations. So copy number variations is where you get, instead of having a perfect 2 copies of every gene, in some people you'll find that one gene has been deleted, or one gene has been duplicated, or even tripled, or inverted in chromosome sequence, so you get chromosome rearrangements of various sorts. I mean, you know, they might be very small rearrangements, ones that we can barely detect. I mean, our techniques at the moment for detecting these copy number variants are pretty crude. And my prediction is that as we get better with sequencing technologies, we're going to find a lot more of these. And already a lot of autism appears to be due to these copy number variants. But also just irregular SNP polymorphisms. Yeah.
Autism seems to be very high in boys compared to girls. Is it something to do with it, maybe CNVs as well?
I don't know that there's any higher rate of CNVs in boys than girls. Actually, understanding sex differences is very difficult. There's hardly a disease where you don't find a difference in prevalence between males and females. You know, some go one way, some go another. What is actually causing that sex bias is I don't think there are any well-worked examples. So, for example, schizophrenia is more common in males, depression is twice as common in females. You know, bipolar, I've forgotten which way bipolar goes, and so on. But you'll find these biases, and also in the systemic disease, in the autoimmune diseases, you'll find biases as well. So, I mean, it provides quite a nice opportunity in some ways because you know, the cases that are going to be most genetic will be where you actually find a familial case in the sex where it's rarer. So if you're looking at disease which is much more common in females, well, we'll say alcoholism, which is got a much higher prevalence in males than females, then you know, you might find bigger gene effects in a female relative of a female alcoholic than in a male relative of a male alcoholic. So people are sort of trying to make use of that. Yeah.
We might have to make this the last question, and then the food's arrived, and we
Getting
encourage
cold.
you to
Okay.
continue your discussions outside. So just the last question.
Yeah.
So in Australia, I'm just getting a neighbour, I think. What are like the numbers of twins and then non-identical twins compared to like the ones who take part in this sort of?
Yeah, well, that's a good question. The rough reckoner which is not so rough actually, is you divide the population by 100, and that's the number of twins that you'll find. That's in European ancestry populations. And in European ancestry populations, about a third of those will be identical, and about 2/3 will be non-identical, and of the 2/3 non-identical, a half will be boy-girl, and the other half will be same-sex pairs. Now that differs between ethnic groups. In Asians, the rate of dizygotic twinning is much lower. It's a quarter of what you see in Europeans. And in Africans, it's about double what you see in Europeans. So there's something like an 8-fold difference between Africans and East Asians in the non-identical twinning rate. Whereas the identical twinning rate is the same between all races. It's about 4 per 1,000, about 1 in 250 births is identical twins, and about 1 in 80 births are non-identical twins. But that rate of non-identical twins is going up quite steeply in our population over the last 20 years for two reasons. Firstly, women are having children later, and the later they have their kids, the higher the risk of non-identical twins. It's about 4 times higher when a woman has a child at 37 than it is if she has a child at 18. Whereas the rate for identical twinning is exactly the same, it's not affected by maternal age. And the other big factor is in vitro fertilisation, which is producing a lot of non-identical twins. So we see those rates changing all the time. But interestingly, I'm right in the middle of negotiations with a government department which would actually enable us to recruit all the twins in the population. And I actually just got the numbers out for Queensland, and there are about 4,500 twin pairs per year born in Queensland. And I said, oh, gee, that's higher than expected. And I went and looked up, who knows what the population of Queensland is? It's 4.5 million. So my ratio is exactly right, it's 1 in 100. Yeah, so about 4,500 pairs per year. Now, into our mole study, we're only getting about 100 pairs a year, so we're only getting a fraction of them. So I'm really excited about it. This is just something that's going on this week. I'm still trying to get access, well, just to get this department to actually mail all those twins and say, hey, please go to this website and enroll in this fantastic study. In which case, I hope we get much, much larger numbers to take part in our studies.
Right, can we thank Professor Nick Martin again? Just before we head out, I just wanted to say this is a SUS event, and if anyone's interested in helping organise an upcoming event or anything like that, I'm Alex, I'm part of the executive. Luke, of course, is part of the executive. Scott, just at the back, no way, Scott, is our president, and Zach's also here as well. So if you have any questions or if you have another idea for a talk like this or anything like that, I really encourage you to come and talk to us. Give us some of your ideas. We're always happy for anyone who wants to volunteer or be part of the community. So yeah.